Cool. Thanks for coming, everyone. The room looks a lot bigger than I thought it was going to be. I look up here, I can see all the faces. And I hope I can keep you interested during my talk here. So I'm going to stick with the grosser side of the fisheries world and talk about parasites. But hopefully,
Yeah, use the microphone.
Yeah.
Or people need to.
Can everyone hear me in the back now?
Yeah.
Okay, sorry about that. Thanks, Randy. So I'm going to talk about parasitic copepods and hopefully on fish that people don't normally eat, rainbow darter and mottled sculpin. They might not be as tasty as striped bass, so hopefully I don't approach you out too much. So the title is Distribution and Ecology of Ergasilus cotti, so a very specific animal described in 1892, and it's obviously found on those two specific animals. And most of this project is was started by a gentleman at USGS. It's started by a gentleman at USGS Ann Arbor, and he retired in about 2003, and he hasn't picked up this project in like 15, 20 years, and he's threw it in my lap. So it's been a kind of a logistical nightmare, but I'm excited to talk about the story of it, more or less. So there's a lot of different forms of parasitic copepods, and they all start out looking like regular planktonic copepods, much like a Cyclops stomacide, normal fish food that you would see in the water, but they metamorphose into very bizarre shapes to have very specific attachment points depending on the fish species that they're attaching to. And this is the animal that I'm going to stick with today. This real kind of weird shape to it, and it's almost like a burrowing, much like a gill lice would be on, say, Pacific salmonids. So they share the similarity in their form to start with, but when they become parasitic on the fish, and only the females are parasitic on the fish in this genera. So the adult females will find a host, and then they will metamorphose to fit their specific needs on that whatever fish they have. So otherwise, they are planktonic copepods, but they're a unique unique linkage between the copepod world and the fisheries world. So they're in the order Cyclopoidea, family Ergasilus, genus Ergasilus. Just to give you guys a scope of where we're at taxonomically, there's been reported 173 Ergasilus species worldwide, and 26 are known in the Nearctic region, and I'm sure that is expanding because there's not a lot of people doing research on stuff like this. There's probably like 5 people in the United States that are interested in this specific animal that I'm talking about today. So I'm going to try to get you guys a little bit interested in the parasitic world. So there's a lot of variation in the genus Ergasilus. They have a lot of different sizes and form and function based off of what fish they're attaching to and where they're attaching to. So a lot of these are gill parasites. This one here in particular is fairly unique because it attaches to the buccal cavity just in front of the gills. Usually they have like a sweet spot they like to hit for some reason. The animal is so weird and specific, and that's more or less why we're studying it, to have a better understanding of such a bizarre animal. So these are confirmed hosts for Ergasilus of New York, North America. And this is just to give you an idea of generalist versus specialist. A lot of these are generalists, and this is mostly Great Lakes species that have been reported. This is a non-native from Japan that was reported in the Great Lakes 10, 20 years ago, and then recently in Oneida Lake in 2018. And then you have the specialists, and there's only a few of them that are known right now, and it's probably because people aren't searching for it, and that's like a big limiting factor. And and it's hard to explain why it's important to do this, obviously for diversity's sake, and if you're transporting one fish to the other side of the country, you're going to take their parasite assemblage with it. So it's interesting to know everything about a fish, including what's living inside it or on it. And these are all ectoparasitic copepods, so it doesn't necessarily live in the fish, it's on the fish. And this is a couple of genetic trees that we generated from another project that we're working on for Oneida Lake and Great Lakes region. And this just is to highlight how taxonomy, for those weird forms, you can use taxonomy obviously to find and ID the animal and tell more about it and more about the fish it's on, but you can also couple that with genetics, which has been really important, and I'm excited to dig into this a little bit more. I'm not a geneticist, but I'm very curious to learn about how some of these things shake out and why they shake out. So let's get into the Ergasilus cotti and why it's so unique. Here's a picture of one right here. It's very small, and it kind of has that opaque color, which makes it very difficult to find unless it has these fairly large eggs. And it's about 4 millimeters total with the eggs, 2 millimeters without it. So it's very easy to miss these things. Very tedious microscopy work. So it's very specialized as that special attachment point. Very narrow host association. It's known just to these two species currently, and it's most highly modified host attachment cephalosome. So once it attaches, it looks like a regular planktonic copepod, and then it has this very bizarre modification to a cephalosome. So here's the goals of this whole study here. So first, to find the distribution of Ergasilus cotti, because it's only known for a couple different places. So I'm trying to expand the geographic known range. The ecology and biology of the Ergasilus cotti and how it relates to the mottled Sculpin and the rainbow darter. And then do a morphological and taxonomic redescription of Ergasilus cotti. The initial description is about a page long, and if you know anything about taxonomy, that's not very helpful, and it doesn't have enough details, and it's very disappointing. So I'd like to add a little bit more detail for future researchers so they're not as confused as I am. So this will eventually culminate to the first detailed report of Ergasilus cotti since its discovery. And this is just to show you a general idea of the populations of mottled Sculpin in the U.S. and then rainbow darter. So this is the geographic region I would like to do, but obviously that's a headache. So I stuck with the Ohio, Pennsylvania, and New York corridors here. And to do that, I didn't collect any live fish. I used the rich voucher specimen resources at a number of different institutes. Pat Hudson, the gentleman before me, utilized Ohio State, and then Michigan, and then Ohio Northern, and then I dipped into some Cornell specimens as well because I wanted to have a broader range. He stuck with just Ohio, and I'll show that here. So this is a map of the timeline of events. This is the original description in 1892, that really short description, and it was on mottled Sculpin in the Alum River Creek, and which is, I think, a tributary of the Ohio River. And in 1940, it was again found by a gentleman named Tidd at Ohio State University on a rainbow darter, and then he found it again on a rainbow darter on the south shore of Erie. And some of these reports, they don't have the GPS coordinates, it just kind of says where it's from, especially this one. This is a survey of the complete parasite community in Superior on different fish. And there's mottled Sculpin here, but it could be in any of these northern bays. And they more or less study the whole northern shore of Superior. So it's kind of hard to pinpoint where that animal is found, but it's arbitrarily in this general area. So past study area difficulties, remember that Pat hasn't picked up this in 10, 15 years now. So I had some difficulties with this thing. In middle school, I used to use these to prop up my drawers when they're a little rickety, but this has been a headache. I think the data is corrupted on it, so I had to actually reverse enter the data, and obviously that causes problems. Maybe I'm not doing certain things correctly, but that's been quite a headache and a hurdle, and I still have it. And if anybody knows how to fix something like that, please let me know. I'm very curious. So this is all Pat's stuff. He found, just a summary stats of what he found. He covered over 1,200 fish. He only found 21 infected fish, only on mottled Sculpin too, so that's very interesting. And he found a total of 89 parasites. So they're not a problem for the fishery, they're a natural occurrence. And in his studies, he found around 4% infection rate with a 4% mean intensity on any fish found to have parasites. So this is the included area of study. Most of these black dots, well, all the black dots are where nothing was found. So I have a couple in New York, Finger Lakes region, and then Pennsylvania had some good hits. And then this is in New York, technically. It's it's in Chautauqua County. It's very close to the border. But I did end up finding this parasite in New York. So that kind of brings it into perspective a little bit. And then I added another 400 or so fish from the voucher collections. And so this is the complete stats. I added quite a few more parasites, and hopefully they'll be submitted into a voucher collection somewhere at some point soon for future reference. And then I tripled the fish infected found, and mainly because I added these other spots in Pennsylvania and then one lone one in New York. And the total sample dates was from 1922 to 2017, and the recovery dates were from 1922 to 2014. So this animal is definitely still around, but in very low numbers. And there's more to learn on it, but I think I have enough data to move forward on a publication or a manuscript at some point soon. And this is just another breakdown of where the mottled Sculpin were found to have parasites, and then the rainbow darter were found to have parasites. Rainbow darter infection was less than mottled Sculpin infection, but not by very much. So I'm not entirely sure how much that tells us if they have a preference of mottled Sculpin over rainbow darter, but there's more data to look at to see if there's any statistical differences there. So we looked at 843 mottled Sculpin total and 923 rainbow darter total, so that's a lot of fish. Here's a timeline of occurrences just in a different format, and I added my earliest finding in 1922, and then the most recent finding in 2014. So this is the short timeline of events here, or long, I guess. And this is the optimal fish size, theoretically, of where these parasites, it's like a porridge scale. It's too hot, too cold, right in the middle. And they seem to like this 40 to 50 range. That's where the highest parasite prevalence was on these fish. And then they can go as low as 25 in rainbow darter, and as high as 52, and as low as 30, and as high as 81. Obviously, with more data, that number will shift a little bit, but it helps me get I can really kind of hone in on an optimal size, and maybe I can get more parasites at a higher clip. So the result so far is just a summary of what I had before. The total number of parasites found, mostly ovigerous adult females, meaning when they have eggs, meaning they're more developed, were observed between May and August, which is pretty common for Ergasilus species. The presence was found between March and September. There's the total prevalence rates. And then I'm going to show you some cool pictures of the parasites here. So you can see them right here with the eggs. I'm sure if you just looked at it, you'd be like, I don't see anything there. On to the next fish. I actually went through a whole set of fish, and I was very discouraged not to find anything. And then I don't know why I did it again, just out of curiosity. I went through them again, and I started finding them. So it was very tedious. Thank you. But it was eventually fruitful, thankfully. Otherwise, I was going to be very discouraged, and I probably would have just picked up another project. So you can see these parasites here, and they have some blue pigmentation on the eggs sometimes, and that's when the knocklet are actually about ready to hatch. And imagine if you didn't have the eggs, they're so opaque, and they don't look like anything when they're in there. They just look like part of the gill. This is the pigmentation I was talking about on the eggs. You can see the cute little knocklet getting ready to go infect our next fish, going to the plankton. This is our Ergasilus cotti who we removed from a buccal cavity. This is just a far out picture. You can see kind of like the egg sac here. And they have very high fecundity too, because they don't spend a lot of time having to move very much. When they're tasked with the fish, they're not spending a lot of energy. They're just gaining sustenance off of the fish. So they're able to be very highly fecund. And this is the weird, bizarre shapes that they generate in their claws, which is a modified second antenna. And they have these big like latches on them, so when I'm trying to pull them out, usually the head will pop off, and that's the most important diagnostic feature, and that's so frustrating. So this is a picture of how they actually burrow themselves into that fish tissue. So this is just excised from the whole gill, buccal cavity, and my colleague was able to stain it, and you can see kind of the outline of the shape of that cephalosome there. So takeaways from results: rainbow darter is a viable host for Ergasilus cotti, and Pat had suggested that it might not be before I picked up, possibly not preferred, and it does in fact show lower infection rates. Ergasilus cotti is present in Ohio, Pennsylvania, and maybe New York. We'll see. I don't know. I think I need more data than just that one point to really say for sure. A more widespread distribution of Ergasilus cotti is likely. The prevalence and intensity depends on season, habitat, and host, and potential other suitable hosts. That Sculpin kind of looks like a goby to me. I don't know if it'll end up in the lakes, but it probably won't be invasive. But it would be, if I was this parasite, I think a goby would be a pretty suitable host. So future plans are to compare mottled Sculpin and rainbow darter distributions and how they relate to each other, the detailed morphological description I mentioned, and obtain genetically viable specimens. I haven't been able to get my hands on any of those yet, but hopefully soon, and then submit the specimens for future curation. There's a reference list, and then the acknowledgements, and thank you all.